Experiment 3A. Aim: to show that plants carry out transpiration with the help of their leaves. Bell jar experiment. Materials required: bell jars, healthy green potted plant, a pot with soil and polythene bag. Theory: transpiration is a process in which loss of water in the form of water vapor takes place from the aerial parts of the plant body. It is classified as stomatal transpiration. The loss of water vapor takes place from the stomata, which are minute pores present on the surface of the leaf. These are surrounded with guard cells. The guard cells control the opening and closing of stomata. When the guard cells are turgid, the stomata open, and when they are flaccid, they close. Cuticular transpiration. In some plants, the loss of water vapor takes place from the cuticle present above the epidermis of the leaf or stem. Lenticular transpiration. Some of the old stems of plants have pores called lenticels. When water vapor is lost through the lenticels, it is called lenticular transpiration. Transpiration rate is affected by a number of factors like humidity, light, temperature, wind, carbon dioxide concentration, and water in the soil. These are the external factors. Some of the internal factors are leaf area and structure of the leaf. Significance of transpiration. It influences absorption of water from the soil. The water absorbed by the plant helps in photosynthesis. It helps in cooling the plant. It leads to wilting of the plant. Excessive transpiration leads to stunted growth. Procedure. The potted plant is watered well. The potted plant is kept on a flat surface. The pot and the soil surface is covered with a polythene sheet. The pot without the plant is also kept on a flat surface and covered with polythene. Both the pots are covered with dry bell jars. They are left undisturbed for about half an hour. Observations. Drops of water are seen on the inner surface of the bell jar, which was used to cover the potted plant. There were no water droplets in the other bell jar. Conclusion. Water vapor which is lost from the aerial parts of the plant condenses, and so water droplets are seen. There is no other source of water in the setup. No droplets are seen on the bell jar, which was placed over the pot without the plant. Precautions. The potted plants should not be exposed to bright sunlight. A healthy plant should be chosen for the experiment. The soil surface should be covered properly with a polythene bag. The bell jar should rest properly on the flat surface to ensure that no air enters.